So what do enzymes do? Are we recording?
Catalyze biological reactions.
How do they do that?
Lower the activation.
See, I did better in biochem last time than before. So you guys have seen this, right? So basically, the transition state energy, or what's it called, the free energy of activation. Like in this example, I like this example. I don't think I showed you this for biochem. This is basically the oxidation of glucose, converting glucose into carbon dioxide and water. Or is that one or the other? That's water down there. Carbon dioxide and water. And to do this, first you have to break the bonds between the oxygen and the sugar, and then you get back energy in return from forming the water and carbon dioxide. And this is just if you broke all the bonds at one point. You have to put that energy into it to break them, and then when they form new bonds, forming the water and carbon dioxide, you get back the free energy. So, huh? You like that, don't you?
Yeah.
I always thought that was easy points in the biochem test, but some people have gotten it completely wrong, which makes me sad. So enzyme commission classification. So there's all these different classes of enzymes, right? We got oxioreductases, transferases, hydrolases, lyases, isomerases, and ligases. So that's the 6 classes that they're labeled in. Oxioreductases, what do you think that involves? Oxidation reduction reaction. So transfer of electrons from one substance to another. An example of that is glyceraldehyde 3-phosphate. Do you know this? Anybody know this enzyme that catalyzes this? Phosphate plus glyceraldehyde 3-phosphate plus NAD yields NADH plus hydrogen 1,3-bisphosphoglycerate. Yeah.
Glyceraldehyde phosphatase.
Not phosphatase.
Damn it.
Because you are adding a phosphate to it. Phosphatases, remember, remove phosphates. But that's not what's going on in here. It's an oxidation reduction reaction. See, you reduced NAD to NADH. So yeah, glyceraldehyde dehydrogenase, GAPDH. For you, I know you guys have heard of GAPDH, right? No? Methods 2? Yes. So that's an oxioreductase, GAPDH. Transferases, it's just you have X combined with Y, and you just transfer X for Z for Y. So it's just transferring one thing to another. Then you have hydrolases. So that's just you hydrolyze some bond, adding water across, right? So here you have A combined with B. Terrible words. And then you have the alcoholic A and then B plus the hydrogen. Lyases are enzymes that catalyze the breaking of various chemical bonds by means other than hydrolysis and oxidation. Example of that is ATP forms cyclic AMP and pyrophosphate. Normally when you break the phosphate bonds, that's a hydrolase that's doing that, right? You add water across those bonds but with cyclic AMP, you cyclize it, you're using the ribose on the cyclic or on the AMP to do that rather than a water. Isomerases, they're just changing the form, right? Enzymes that catalyze the structural rearrangements of isomers. Can you give me examples of that? Triose phosphate isomerase. You don't remember that? Glycolysis? There's 2 isomerases in glycolysis. So one is that, the other, converts glucose to fructose. They both work via an enediol intermediate. You guys remember that? Probably not. Huh?
Barely.
I told Aaron I'd wait. I didn't. she's really late now. Oh well. Huh? Or a vulture.
Buzzer. We don't have vultures, we have buzzards.
Turkey vultures. That's what I've always called them. They look like turkeys.
They don't look like turkeys.
They do. If you're driving by, you see them on the side of the road real quick, it almost looks like a turkey.
Turkey buzzards, they're turkey vultures.
Yeah, the turkey vultures is what I've always heard.
Let's find out.
Okay, let's take a vote.
I don't think they look like
I think buzzards is another layman's term for them, but
turkeys.
It's also known as turkey buzzard. The very first line, the turkey vulture, also known as the turkey buzzard. Sorry.
okay. And then ligases, we've used ligases, right? What do they do?
Ligation.
Fill in the gap when you have a nick on DNA. So, ligases, 6 classes. And then you have the EC system, which it's represented by 4 digits. You have the main group, followed by the class, the reaction catalyzed, and then the acceptor or subclass, and then a serial number. So that's these numbers. I'm not going to make you memorize
So what exactly
any of these.
does EC stand for?
Huh?
What does the EC stand for?
Enzyme Classification System? I don't know. Look it up. What's it say, Andy?
If I type in EC, I'm going to get like 3,000 responses. Hold on.
Type in EC and enzymes. Enzyme Classification, maybe. And then IUB, you get a systematic name, European Commission on enzymes. Is that what it is?
EC number, Enzyme Commission number.
Don't worry, we just started. Just reviewing a little about enzymes. So these are some of the examples. You have your EC numbers like trivial names, lactate dehydrogenase. You guys remember that? Lactate dehydrogenase. The EC number is 1.1.1.27. So it's a dehydrogenase, right? So it's going to be one of those oxioreductases, which is number 1, right? So 1.1.1.27 and huh?
I'm just very distracted.
Sorry, it's okay. But I'm not going to make you memorize these. You just got to be aware of what the numbers mean, basically. So. Yeah. So the first number is the main group, which in lactate dehydrogenase, of course, is an oxioreductase. Second one's the reaction catalyzed. Third is the acceptor or subclass, and the fourth is the serial number. So 1.1.127 lactate dehydrogenase. So, and then the systematic name is L-lactate NAD oxioreductase versus 1.1.1.49 is D-glucose-6-phosphate NADP oxioreductase, and so forth. So I guess they all share NAD, right? They're all those two are oxioreductases. The acceptor is going to be NAD, and the reaction catalyzed is dehydrogenase, right? So pretty simple, maybe not. So onward. So enzymes are biological materials with catalytic properties, right? They're proteins. And there's 2 different classes. You have some that are always on, right? That's what this means. Can somebody say that? Constitutive? Constitutive? Constitutive. The other is inducible. What does that mean?
It can be induced.
Yeah, the cells turn it on, right, in response to something. Changes in enzyme activity in body fluids can be a valuable diagnostic tool. Anybody have any ideas of some enzymes that you measure for diagnostic reasons? Yeah, what would that tell you? Yeah, I think it does. What? Yeah. So different tissues or cellular material contain different quantities of and types of enzymes, right? So because lactate dehydrogenase is basically expressed in every cell, right? So how would it tell you you're going to have a heart attack?
Kind of like our cardiac enzymes.
But don't you say that in hemolysis too?
Huh?
Don't we say that in hemolysis too?
Hemolysis? Yeah, you probably would, yeah. You definitely would.
When you said
So there's got to be some way to distinguish what tissue it came from, right?
Well, yeah.
How could you do that? Why would it be different in different tissue? And what would be different about the enzymes to make you able to distinguish them?
Creatine kinase. You know that.
So we'll continue.
Troponin. Oh, that's right. Troponin levels. Where's troponin?
Yeah, is troponin really an enzyme?
No, when you do cardiac enzymes, you remember troponin.
Yeah. So proteins, they're, between 500,000 Daltons. What's primary structure? Right, amino acid sequence. Secondary structure is going to be alpha helices, beta sheets. That's interactions between two locations on the protein chain. Tertiary is the folding of the chain, and then the quaternary is two or more polypeptide chains together. What are coenzymes? People always miss these. So coenzymes are non-protein components of the enzymes, right? So can you give me some examples of the coenzymes? We already talked about one. Huh? Coenzyme A, yes. That one's got the coenzyme in the title.
Acetyl-CoA.
NAD, NADH. My favorite's TPP. Right? You guys like that? I made you draw out the mechanism for that. Huh?
I thought it was an extra question.
It was. I made it required this year, though. So I made everybody had to draw all the mechanisms out on their own time and turn them in. But I think it improved their test scores. Maybe. So, cofactors. Vitamins or contain vitamins, so thiamine is TPP. Cofactors, also like NAD is a vitamin. Actively involved in the catalytic reaction. They can be covalently bound to the enzyme, which makes them a prosthetic group. And then if you have one that's covalently bound, can you think of one that's covalently bound to an enzyme? It has that tether. I don't think TPP is covalently bound, is it? Huh? It's got the handle, right? Because it's got the A and P. Huh? Yeah, which one swings? There's two that swing around. Biotin. And lipoic acid. Yeah. So a holoenzyme contains the prosthetic group that's covalently bound and the protein. Apoenzymes are only the protein. So because they're proteins, things like pH are going to affect their active sites, right? As pH goes down, what happens? You increase the concentration of protons, which is going to protonate some protonatable groups, right? Make them more positive. So similarly, as the pH goes up, you'll lose protons. So it's going to alter the structure at the active site and possibly killing the activity. So you're going to want to, when you're doing this in the lab, you need to use proper buffers at the right pH. We're going to be doing alkaline phosphatase. What kind of pH do you think we're going to need?
Probably alkaline.
Yeah, exactly. So, and then cofactors and activators, you're going to make sure you have all of those. Which, for the one we're going to be dealing, alkaline phosphatase requires activators, and that's why you can't use EDTA blood or citrate blood, because it'll tie up the metal ions required for that. Yeah. Temperature, we also have to use the right temperature, right? Because they're proteins. Normally when you have an enzyme, or or not an enzymatic action, a non-enzymatic action, as you increase the temperature, you're going to increase the rate, and that's because you're making the size of that activation energy smaller, but it's because you're boosting the average energy of all the substrates. Whereas with an enzyme, as you increase the temperature, you will increase the rate to a point at which you start unfolding that enzyme, denaturing the enzyme, and then activity will drop off. So, functions and characteristics of protein isoforms. So this is where we get to why you can identify certain lactate dehydrogenase from being from certain tissue. It's because it's the isoform or isoenzyme, right? And what happens is different molecular forms of a specific protein, okay? And it could be produced from different but related genes, right? It can also be produced from the same gene but represents different RNA splicing. You know what I'm saying? So you get alternative splicing can result in a functional change on that enzyme. Also, single nucleotide polymorphisms can result in different enzymes. And 90% of the isozymes, though, result from post-translational modifications. What kind of post-translational modifications are we talking about? Glycosylation is one. Are you reading the thing? What other kind of post-translational modifications are possible?
Translational?
Yes, translational. What kind of accent does that sound like?
That sounds like nothing but confusion.
Okay, good. That is my brain when I try to speak. You could look.
Phosphorylation?
Yes. Fragmentation, right? Give me an example of an enzyme that has to be fragmented before it works. Huh? Yeah, but insulin's not really an enzyme, but they use that in the book, don't they? Biochemistry is an example. But it's a hormone that has to have fragmentation. But there are enzymes, a whole group of enzymes. You guys are studying a class that really studies one of them, so you should know all of these. Huh? You were doing the test for it the other day. PTT. You know, the activated PTT, you activated it by, letting it sit there and get digested first, right? Or I don't know how that works. Don't listen to me. I was just guessing that's what the activated prothrombin time is, right? You pre-digest the prothrombin to make thrombin, so you get an idea of like the total amount that's there, right? Does that make sense?
No, yeah, that's right.
Okay. Yeah, so prothrombin, it's called prothrombin because it's inactive, it's in its inactive form. You have to cleave it with a protease. I forget which one does it. You guys know the cascade?
Not well.
Because it's another one of the blood factors in the cascade. Cleaves prothrombin to thrombin, and then what does thrombin cleave? Fibrinogen or?
Is it factor 7?
Oh, I'm not comfortable enough to get it wrong.
I'm not grading you on the blood stuff. That's Dr. Smith.
Well, it's been like over a year.
Yeah. So, isozymes, genetic variations that arise from multiple gene loci or one gene loci. So, or from allelic genes at a particular locus. All isozymes possess the ability to catalyze the enzyme's characteristic reaction, so they share the same EC number. So those lactate dehydrogenase, no matter what isozyme it is, it still shares that same EC number. What differs is their ability can be inhibited by specific compounds. So you can use specific inhibitors to knock down the activity of one isozyme and measure the activity of the others. The KM, do you guys remember what the KM? We'll talk about that again in a minute, but I'm not going to go through the... Yeah, or should I? Is that fun for you guys? No, I'm not going to test you. So some can react with different substrates. So you have different substrates that aren't their natural substrate that'll react with them. And then may also differ in some physical properties, such as the PI. What's the PI? Isoelectric point, which is, net charge is 0, right? That's the pH at which the protein has no net charge. Heat stability. So in our lab, we'll be differentiating isozymes via heat stability. So the bone alkaline phosphatase is inactivated at 56 degrees. We're going to incubate our samples at 56 for half an hour, try to kill off the bone alkaline phosphatase in the sample so we get an idea of total alkaline versus just the bone alkaline. And then immunological reactivities. So you can use antibodies to block activity if there's against, or you can like precipitate them out of solution with antibodies, specific isozymes. Or you can purify them. There's enzyme assays where you can run the sample through an ELISA where you have a capture antibody that's only going to recognize one specific isozyme. And then you do the reaction with that enzyme bound to the antibody on the solid surface of the ELISA. So structural basis for isozymes. Amino acid sequence of isozymes are typically homologous. So they're typically very similar, but doesn't mean they're the same. They're just homologous. Small changes in amino acid sequence can result in differences such as catalytic properties, active site structures, PI charge distribution, hydrophobicity patterns, pH optima, and Km. And this is an example of isozyme selective inhibition of NOS in both of these. And it points out a couple, there's just a couple amino acids here. So, which causes a conformational changes in the conserved glutamine and arginine side chains prevented by bulky isozyme-specific amino acids distant from the substrate binding pocket. So this is inducible NOS. The inhibitor tail projects upwards. Do you see an inhibitor tail? So this is the inhibitor. See that? I don't know. Don't worry about too much about this. But it's just to show you just a couple changes in distant amino acids totally changes the structural conformation at the active site, which can alter the inhibitor for it. So, I'm dying of thirst. So genetic basis. So you can have multiple gene loci. So here you have ADH1, ADH2. And they're going to have different loci on the chromosomes themselves. And that makes them isozymes, which is slightly different than allozymes, right? You guys remember molecular biology?
No.
No?
It's like 3 years ago.
Or some of you guys are in it now, right?
Yeah.
Do you guys talk about
I'm sure we did.
stuff?
What did you say?
Allozymes. So if you notice, you have an S allele and an F allele, and they're different ADH1 enzymes because they're not quite the same. They're homologous, but they're allozymes instead of just isozymes. And because they have multiple gene loci, then you can get different expression patterns in different tissues. And this is an example of retinaldehyde dehydrogenase expression, retinaldehyde dehydrogenase 3 versus retinaldehyde dehydrogenase. That's 1 versus 3. Did I say 3? You know me, I'm backwards. So retinaldehyde dehydrogenase 1 versus retinaldehyde dehydrogenase 3. And it looks like you get a lot of retinaldehyde dehydrogenase 3 in the eye tissue, right? Is that eye tissue? Or olfactory? Versus other tissue in these embryos. And it's based on the different expression in different tissues. It's going to give the enzymes a different function for the needed tissue. So, post-translational modifications, we talked about thrombin and prothrombin. Prothrombin is activated into thrombin via proteolytic cleavage. Protein degradation, well, you know, there's a modification, right? When you get rid of that, you're not going to have as much activity. Covalent modification, redox covalent modifications, acetylations, methylations, phosphorylations, dephosphorylations, and N and O glycosylations. All change the activity. And here's a figure showing you all the different modifications that can occur.
What is N and O glycosylation?
So N and O glycosylations are where you add sugar molecules to like groups with an amine side chain or groups with an alcohol side chain.
Oh, okay.
Yeah, such as serine or tyrosine.
Okay.
Is that right though? Yeah. And then N is going to be, lysine or what's the other basic? Arginine, right? Arginine, lysine. That's right. Huh? I should know. I do know.
He's a smart guy.
Well, then why are you making me question that you do?
Well, 'cause I want to see if you know.
I didn't know. That's why I asked.
So microenvironmental distribution. Isozymes have functional significance, differential expression in subcellular organelles, and different structures and net charges influences interaction with other charged molecules within the cell. ALP isozymes are attached to the membranes via carboxy-terminal glycan, phosphadiol, and nitosol anchors. Do you guys remember those? Anybody know what other protein has a GPD anchor? Prion protein? Or, yeah, that's prion protein's phosphanidazole, right? I'm pretty sure. Anyways. So macroenvironmental distribution. So for example, lactate dehydrogenates, we know it converts lactate to pyruvate or vice versa. There's 4 polypeptides of 2 types. And you have one is the M type peptide, the other is the H. And basically you just mix it up with the different 4. It's a tetramer. So LD1 isozyme has 4 the H form. LD2 has 3H and 1. What do you guys think the M and the H stand for? We're talking about differential distribution in the body. Any ideas? What kind of tissue starts with an N?
Muscle.
Yeah. What kind of tissue starts with an H? Heart. So LD1 is found mostly in the heart. LD5 is found mostly in the muscle, right? So you have LD1, LD2, H3M, and it just has to do with the different amounts of M and H peptides. LD3 is a quatermer of 2 H's, 2 M's, and so forth. LD1 mainly converts lactate to pyruvate. When do you want to do that? Well, yeah, when you have lots of oxygen around, that you can turn that pyruvate into sugar and burn it oxidatively. There's no reason, normally when in the muscle, right, you convert pyruvate to lactate because you're working so hard, you don't have enough oxygen for the electron transport chain. So you got to regenerate that NAD from the NADH. It has to go somewhere, that hydride. So you give it to pyruvate to make lactate, kick it out, and your liver will convert it back into sugar. And that's the major form in your skeletal muscle, LD5. So this is the reaction. Again, pyruvate to lactate for anaerobic glycolysis. LDH is elevated in myocardial infarction and blood disorders. So which type you think you're going to test for in myocardial infarction? Yeah, LD1. And it's a tetramer protein made of 2 types of subunits. This is the same thing. I should have just had this first, right? It exists in 5 different isozymes with various combinations of H and M subunits. And there they are again, right, in a big scale. So interestingly, the liver you find also LDH5, but often what's the Cori cycle? You're converting pyruvate to sugar. So you convert it back to lactate first. So, anyways, creatine kinase. So CKMM's it's the predominant form in skeletal muscle and cardiac tissue. CKMB is present mainly in the cardiac. So you'll find both CKMM in the cardiac and the skeletal muscle, but CKMB, you get a much higher concentration in the cardiac. And this just shows you your cardiac markers, right? So you have, this one is cardiac troponin after unstable angina. No, that's what this is. This one's the cardiac troponin after AMI. And then CKMB after AMI. So you get a slight delay, and then it maintains for a couple days raised CKMB. Creatine kinase, it's where you create phosphocreatine or phosphocreatine. And what's that for? That's the serves as an energy reservoir during muscle contraction, right? That donates it to ATP or ADP to generate ATP again, catalyzed by creatine kinase. Skeletal muscle contains the M subunit, and the brain contains the B subunit, and there's 3 different isozymes are formed. You have CK1, which is BB, and it's present in the brain, CNS tissue. CK2 is MB, and that's mainly in the heart. And when that's elevated during acute myocardial infarction. And then CKMM is skeletal muscle, or the myocardium also. So another one you can differentiate from diseases. Alkaline phosphatase is the one we're going to be testing for. So what do you think that does? Phosphatases, somebody already talked about it today. Phosphatases remove phosphate groups. Kinases add phosphate groups. So in this example, we're adding phosphate to creatine via creatine kinase. Alkaline phosphatase removes phosphate groups. So it's a group of enzymes, and it has maximal activity at a high pH, hence is why it's got the name alkaline phosphatase. It's widely distributed through the body. High levels are seen in the liver, bone, placenta, and intestines. And it's useful to assess hepatobiliary and bone disease. In hepatobiliary obstruction, hepatocytes lining the biliary ducts induce ALP synthesis, so you'll see raised ALP enzymes if you have hepatobiliary disease, like a bile duct obstruction. High levels of ALP is also indicative of extrahepatic obstruction rather than intrahepatic obstruction. And then in bones, the enzymes derive from osteoblasts. Which ones are those? Remember, there's osteoclast versus blast. So the blast add, right? So increase in bone disease like rickets, you'll see them, increased ALP. What's the matter?
He's making faces.
You both were making faces.
That's just my face.
So this is the reaction we're going to be doing in the lab, right? So remember, this removes phosphates. So we have paranitrophenol phosphate as our artificial substrate. Paranitrophenol phosphate does not absorb light so much anyways. So if we treat it at about pH 10.3 with ALP and magnesium, it converts it to paranitrophenol, which is a colorless form, and then it gets rearranged to paranitrophenol, the quinone form, and it's very bright yellow, and it's real easy to read. So it's a nice substrate that you'll see in a lot of different reactions. And then we read the color at 405. The activity for bone isozyme can be estimated by heat treating the sample at 56, and that's what we're going to do. So we'll read total ALP, get an assay for a total ALP activity, and compare it to the heat treated sample and see if there's a difference, and then you can calculate how much of the total was the bone ALP activity. Measurement of ALP before and after heat treatment gives the measurement of bone ALP. We're just going to have 2 samples, and we'll heat treat one, and then we'll incubate both at 37 with the substrate. Acid phosphatase also removes phosphate groups, but it doesn't under acid conditions. At pH 5 to 6, it's present in the prostate gland, liver, spleen, and the main source of acid phosphatate is the prostate gland, so it can be used as a marker for prostate disease. Amylase, what does this do? Yeah, it breaks down sugars. So it's a digestive enzyme. You see it in your salivary glands produce it, but also your pancreas You're going to see it in the blood if you have pancreatitis, acute pancreatitis. And it's used as a marker to detect pancreatitis and appendicitis. What about ALT and AST? Yeah, they're elevated in liver disease, right? But they're transaminases. So aspartate aminotransferase takes alpha oxoglutarate. Anybody else know what the name of that is? Alpha oxoglutarate? There's an alternative name for it. It's in the TCA cycle. Alpha ketoglutarate. So I don't know why they call it alpha oxo. I guess because it has that oxygen on the number 2, on the alpha carbon. So alpha ketoglutarate and plus L-aspartate, it's an aminotransferase, so it just takes the amine group from the L-aspartate and transfers it to the alpha ketoglutarate to make glutamate, and then you're left with oxaloacetate. Same thing with alanine aminotransferase. You take alpha ketoglutarate, takes the amino group from L-alanine and transfers it to alpha ketoglutarate to make L-glutamate and pyruvate. And those reactions are obviously reversible too. So we see ALT and AST, they're most abundantly present in the liver. So these are the enzymes you often hear about being your liver enzymes. So if your liver has been damaged, you'll see liver enzymes spilling into your blood due to necrotic tissue, and you'll have raised ALT and AST enzymes. So measurement of these transaminases is useful in liver disease, and in viral hepatitis, the enzyme levels are increased 20 to 50 times above the upper limit of the normal range. Alanine transaminase increase is specific for liver damage involving hepatocellular damage, and then aspartate, or AST, is moderately increased in muscular dystrophy and acute myocardial infarction also. So things to be aware of. And this is just a graph showing you viral hepatitis and the time course that you see ALT and AST elevation, followed by bilirubin elevation. And then these are levels of enzymes we see in myocardial infarction again. So we have CKMB, which is creatine kinase, right? MB is the muscle isoform plus the brain isoform. That's what we see mostly in the heart, right? AST, aspartate aminotransferase, right? LDH, lactate dehydrogenase, and HBDH. What's HBDH? I don't know. HB dehydrogenase. So clinical significance. Oncofetal tumor markers include alpha fetoprotein and creatine kinase BB. Huh? Hydroxybutyrate. Ah. And what's that for? That's a ketone body, right?
It occurs in higher levels of heart muscle tissue, and therefore it's more sensitive and specific in diagnosis of myocardial infarction.
Yes. You see how they're raised quite a bit in the days after, or starting off just hours after myocardial infarctions. So muscular dystrophy alters the distribution of aldolase, lactate dehydrogenase, and creatine kinase. Remember what aldolase does? That was again glycolysis, wasn't it? And you split that fructose into the 2 3-carbon sugars. Right? Is that right? I'm not sure. I'm pretty sure though. And then tumors increase placental, intestinal, and germ cell alkaline phosphatase. So, factors affecting detection in the blood include Altered rates of production. If you're not, you could alter the rates of production, which is going to alter the amount you detect in the blood. Baseline concentrations result from normal cell death. All these tissues have normal cell turnover, which are going to leak a normal small amount of enzyme into your blood. That's why we have the reference ranges. When we see those elevations in reference ranges, that tells us there's some kind of disease process going on, which is what that says. So, Michaelis Menten constants, right? We were going to talk about that. Basically, the Michaelis Menten constant is the concentration of substrate at which you get half your Vmax, right? Does that sound familiar? And it's basically, it's analogous to a KD for the substrate for the enzyme, right? So it's basically the smaller the Km, the higher the affinity for the substrate and the faster the reaction will be. And basically it expresses a relationship between the reaction rate and substrate concentrations. So, and we know this, the enzyme kinetics, I've done this before. I'll just skip through it so you guys don't want to see the whole thing again, right? Kinetics. And then you finally get Michaelis Menten equation down there. But basically, what's special about the Michaelis Menten equation, Michaelis Menten kinetics? It's represented by this graph, right? So there's like two different parts to the graph, right? And this is important for both measuring enzyme activity in blood samples and also understanding how drugs get metabolized in your body. So it has to do with the kind of kinetics, right? When the enzymes are saturated with substrate, you're only going to see one velocity reaction. You basically have approached the Vmax. The enzymes are saturated, so you can't get the enzymes to work any faster. They're working as fast as they can. The only thing is the diffusion of the substrate on and product off is what slows that down and how fast the enzyme can work. So this is all, what order kinetics? No, zero order. Zero order kinetics. And that just means no matter how much substrate you put in there, you're not going to change the rate at which that metabolism occurs. The rate's not dependent on the substrate concentration. First order is here, and that's where your rate changes based on your concentration of substrate. As you increase the substrate concentration, the rate of the reaction increases a lot. Whereas when in the zero order part of the curve, it doesn't. So that's the two things to consider. There's two parts of this curve, a zero order curve and then a first order curve. So if you're trying to measure enzyme like activity in your blood, what part of the curve do you think you want to be on? Any guesses? No. It's a 50-50 shot each time, and each time you guys missed. So if you're trying to measure how much activity is there, so the Vmax is only going to change when you add more enzyme to the mixture, right? So if you saturate the system with substrate, you should only be reading the Vmax. So the more enzyme there, the higher the Vmax will be. The less enzyme, the lower the Vmax will be. So you're just measuring the activity of that enzyme, so you want to saturate it with substrate. So we'll be saturating our ALP enzymes with the nitrophenol phosphate. Paranitrophenol phosphate. I knew there was another P in there somewhere. Paranitrophenol phosphate. And that way, we'll just measure all the enzyme that's in there should be saturated. So if there's more enzyme in the blood, we'll get more activity. If there's less enzyme, we'll get less activity. And that's how we measure the activity. Okay? So we can take that Michaelis Menten equation, and you know, if we do the inverted equation, we can get a graph, like a linear graph, where we have 1 over V max is the axis, I mean the intercept, the y-intercept, 1 over V max. Negative 1 over the Km is the x-axis. And then the slope is going to be basically what? The rise over run, so that's so it's going to be your Vmax divided by your Km, right? Is that right? They don't have it up there, do they? So enzyme inhibitors. So the reversible versus irreversible. Reversible inhibitors interact with an enzyme via non-covalent associations. So you can drop the concentration away and they'll dissociate and eventually won't inhibit the reaction. Irreversible inhibitors interact with an enzyme via covalent association. So a lot of antibiotics are dead-end inhibitors to enzymes that bacteria produce. They just covalently bind to the enzyme and kill it. Classes of inhibition. Competitive inhibition, the inhibitor binds only to the enzyme and not the enzyme substrate complex. Remember, it competes for that active site. So you can overcome the inhibition by increasing the concentration of the substrate. Non-competitive inhibition, the inhibitor binds either the enzyme or the enzyme substrate. And if it's perfect non-competitive, which really might not exist, it might. But if it's perfect, it binds to both enzyme and enzyme substrate complex with the same affinity, right? But typically there's going to be a different affinity. Uncompetitive inhibition, remember, it doesn't exist. That's the inhibitor binds only to the enzyme substrate complex and not the enzyme. And it's hypothetical, it's never been documented in a real case, but makes for a useful contrast. This is just a little reminder of competitive inhibition. So inhibitor and substrate compete for the same binding site, the active site, the substrate binding site. So as you increase concentration of your substrate, you'll outcompete the inhibitor and you'll be able to attain Vmax again, right? So your Vmax, your maximum velocity. So you're going to be able to get the Vmax again with competitive inhibition. Unlike non-competitive inhibition, basically, it's when you're bound to the substrate, it's basically like taking that enzyme out of the system, so it lowers your Vmax. Doesn't really change your Km in pure non-competitive inhibition. And these are the charts again. So you have competitive inhibition, Vmax is the same, so the y-intercept is going to be the same. Non-competitive inhibition, Km is the same, but the Vmax is decreased. So 1 over Vmax will be larger than the 1 over Vmax for the non-competitive one. And then uncompetitive, the two have the same slope basically. So I'm not going to talk much about non-competitive. And this is another figure. I think this figure is from your book, right? And then this figure is from a different book. And not a big fan of this figure. So measurements of enzymes. There's 3 phases, right? We have a lag phase, a linear phase, and a substrate depletion phase. And what happens is the lag phase is when it's just getting mixed. There's a little bit of lag before the reaction. Then you have a linear phase where you have a constant production of product. And then the substrate depletion phase, you're starting to run out of substrates, so you're not going to produce any more product. And that's how it works, right? So we want to measure during this linear phase probably. And there's 2 types, right, of reactions we can use. We can use the endpoint, or we can use, a time course. Time course is probably the most accurate. So, because you're just going to measure the rate during this linear phase. But if you have plenty of substrate and you've really saturated the system with substrate, you should never reach that substrate depletion phase. But if you let the reactions go for too long, eventually you'll deplete the substrate and you won't be able to tell the difference between samples, right? So you won't get an accurate reading. So, and enzymes are not directly measured. So enzymes are commonly measured in terms of their activity, right? You always hear international units or IUEs, or something like that. So we don't measure the molecule. What do we measure molecules with? You'd measure it with an ELISA or a Western blot. And that's just like an immunological quantification, right? So that wouldn't tell us if the enzyme's active. It could be one of the zymogenic forms where it needs to be cleaved to become active, or it could have some post-translational modification altering its activity. So that's why we measure the activity, because that tells us the active form of the enzyme. We're not measuring the molecule. We measure how much work it performs, its catalytic activity, and the rate at which it catalyzes the conversion of substrate to product. And that activity is a reflection of its concentration. It's proportional to concentration, right? So photometric measurements of activity, we have the enzyme plus substrate yields the enzyme substrate complex and then yields the product. So it can be tested in either way. We could have a substrate that absorbs the light or a product that absorbs the light. So we can measure the increase in product or the decrease in substrate, or if there's some kind of coenzyme necessary, what's one that's popularly used, which happens to be, I guess it could be a product or also, but NAD, right? NADH, the reduced form and the oxidized form of NAD absorb differently. So increase of altered coenzyme. If the substrate and coenzyme are in excess concentration, the reaction rate is controlled only by the amount of enzyme there, that enzyme activity. And this is the NAD, the coenzyme that's often used in a lot of these tests. For a lot of these, like not alkaline phosphatase, but for the oxioreductases, you'll see NAD used a lot. And also for those aminotransferases, maybe not. Anyways, you see the reduced form of NAD absorbs at about 350, whereas the oxidized form does not. So you look at the change in concentration of those. And this is just an example. As changes the substrate concentrations, the rate of the reaction. So we have the rate of NADH consumption and NADH concentration. So you have decrease in NADH as the course goes on for that reaction. So you can also, some tests use coupled enzyme reactions. So some enzymes utilize other enzymatic reactions in a sequence of reactions, and these other accessory enzymes are not being measured. They're part of the test reagents, right? So the auxiliary enzymes are used as reagents and added in excess so they don't become rate limiting factors. That's the important thing. You don't want any of the reagents to be rate limiting of the reaction you're trying to measure, because then you'll be measuring the limited rate rather than the complete activity of the enzyme you want to measure. So all enzymatic reactions in a coupled assay must be performed at, remember, zero order kinetics, and that's that flat part of the Michaelis Menten curve where concentration of substrate does not affect that velocity or the activity of the enzyme. This is an example of a coupled enzyme assay. So you have substrate 1 plus enzyme 1 makes product 1, which will then react with enzyme 2. And this is what we want to measure. This is an auxiliary enzyme. Makes a product 3, which reacts with another auxiliary or the indicator enzyme. And we'll read where do we say here? We read either the consumption of P2 or the production of P3. So, and they do that because not all of the initial products are going to be easily measured. And you want something in the lab that you can easily measure with light, typically. So enzymatic reactions are measured in terms of rate at which the substrate is converted into product, and this can be done 2 different ways, and I already talked about this, right? Fixed time assay, where you just have 2 points. Substrate, has to be zero order kinetics. The substrate concentration is measured at a set time intervals to determine enzyme activity. A slight delay from the beginning of the reaction to the maximum velocity, and that just accounts for that lag phase because you won't be getting the true enzyme activity during that lag phase. So extreme elevations in enzyme activity may deplete the substrate between measured intervals, so that's one thing you got to be concerned about is that substrate depletion. How's that going to affect the measured activity if you have substrate depletion? It'll make it seem like it has lower activity than it actually has, right? You'll really have higher activity than what you're measuring due to you're running out of substrate. Oh, and I have it right there, don't I? You should have just answered it. You would have looked smart. And this is a figure showing you that. So if you just do this linear reaction, you take the time point here, it could be due to substrate depletion when your real rate is really fast, right? If you had plenty of substrate at this time point, you really would have been like up above L or O. So. Better thing to do is a multi-pointer continuous monitoring, aka a kinetic assay, where you just read the change in absorbance every second. Some machines can do it. You can't do it truly continuously, right? But you can, our plate reader will read it, but it has to read every time. So it takes, think about a minute, 30 seconds to read all of them, and then start again and read all of them. And we can do kinetic assays with it. So continuous measurement of substrate and product concentration are recorded by the spectrophotometer of an automated analyzer. Substrate depletion is detected because the analyzer is always looking and will see that dramatic change in activity. And you just measure the activity by taking that slope before you get to that substrate depletion phase. So this is measured correctly before substrate depletion, and with this linear assay, we'd only be measuring this where we don't need to worry about the lag phase because we'd see when that occurs, right? And this is another comparison of 2-point versus multi-point. Pretty simple. So enzyme activity is measured as a function of the change in absorbance. So what does that tell us? Remember the molar absorptivity? If you want to convert it to international units, we'll have to figure out the molar absorptivity of the product that we're measuring. So remember enzymes that are measured in terms of activity, how much work is getting done, and how fast a chemical reaction can occur. A large change in absorbance per time indicates the reaction is occurring at a rapid rate. And to calculate enzyme activity, it's not a measure of concentration. It's measured how fast they convert substrate to product, and it's measured in how fast they work. The common unit is the international unit, which is the amount of enzyme that will convert 1 micromole of substrate to product per minute under defined conditions. So we're going to be measuring absorbances, and then there's a certain time point that we measure, and then we can calculate the concentration in our assay of product that'll be produced via its extinction coefficient that we'll have to calculate also. So these can change, right? Extinction coefficients will change based on pH, temperature, and the substrate itself. And this is a simple little equation that you'll use basically. So you have the change in the enzyme activity in international units per liter is equal to the change in absorbance divided by the number of minutes that reaction has been taking place times 1 over the extinction coefficient, or molar absorptivity rather. The molar absorptivity times the path length, and then total volume of your reaction divided by the sample volume. So that's basically what? The dilution factor. And then times 10 to the 6. And that just converts, moles into micromoles, right? Right there, yeah. So simple to do. And an example of that is NADH to NAD, increase or decrease NADH concentration in a solution will cause the absorbance to change at wavelength 340. So we have an absorbance of 0.5 times 10 to the minus 3 molar NAD solution is placed in a 1 centimeter light path, has an absorbance of 0.311. So we can calculate the molar extinction coefficient, or the molar absorptivity, and we calculate it to be 6.22 times 10 to the 3 moles per liter. Each chemical substance has its own unique molar absorptivity. So this is just like a review of our chapter 3, right? But we need to know this to be able to calculate the activity. So enzymes differ in their ability to react with different substrates. There are various enzyme specificities. Absolute specificity, that just means that, you know, it only binds to one particular substrate. There's a group specificity, which catalyzes reactions of a particular chemical. So alkaline phosphatase just reacts with general things that have phosphates added to them and removes them, so it's not really specific. Bond catalyzes reactions of particular chemical bonds, and then you can have stereoisomerism. That'll catalyze specific stereoisomers. And we've already talked about this, right? Factors that influence enzymatic factors. So first order kinetics, the enzyme concentration exceeds substrate concentration. As the substrate concentration increases, the reaction rate increases. The reaction rate is proportional to the substrate concentration, and it's not a good place where you want to measure enzyme activity. But I was going to do that therapeutic drug monitoring. For the majority of clearance of drugs, it's going to be first order kinetics, right? Do you know why? Because you're going to contain, most of your enzymes are in your liver that metabolize drugs, not all of them, but there's a set amount of that enzyme. So the majority of drugs we take are at such low concentrations, those enzymes are never saturated. Can you give me an example, one that's different, that we often saturate the enzyme system that metabolizes it? Ethanol, yeah. So we take so much ethanol, we always saturate that. So that's an example of a drug that we have zero order. That's why they say 1 beer per hour is about what you metabolize. And it doesn't change no matter how many beers you drink or how little you drink. It's always your liver metabolizes the same amount, the same mass of ethanol per hour. Huh? So, but this is where we want to be, right? Zero order kinetics to measure enzyme activity, and that's the substrate concentrations in excess. That's where we are when we drink lots of beer. We've made the alcohol dehydrogenase saturated. Substrate saturates enzyme. All enzymes react with the excess substrate. The chemical reaction rate then goes to its maximum velocity, or Vmax. Reaction rate's dependent on the enzyme activity, or how much enzyme is there. Enzymes are not measured, but activity. And zero order conditions are suited for enzymatic reactions. And I've got this graph again. And then this is the figure from your book. And this is another figure from your book that kind of, I don't know if it's a good one, has your E's. Those are your number of enzymes that you have. For a first order reaction, you have plenty of enzyme around, so that activity is going to be based on the amount of substrate. If you add another substrate, it'll boost the activity a little because you're able to use more of the enzyme. Whereas zero order reaction, it's constant. All your enzymes are burning those substrates through as fast as they can. And it's only the activity is only dependent on the number of enzymes there.
What's the SE stand for?
The SE, so that's an enzyme that's bound to a substrate.
I'll get that ES.
Same thing, it's just bound to the back of the substrate. Or the back of the enzyme. So, I don't know, the book did it that way. I think it's supposed to represent the same thing. They could have made better figures than this, I think. Maybe that's a allosteric regulation of that enzyme. Just joking. Enzymes are not measured. Didn't I just have this? Yeah. These are good. What? Did I have these a couple times?
It's written differently, but it's the same thing.
Okay.
Like the other slides.
Yeah.
So what does that mean? Probably know from
Yes. So, okay. And that's it for this, I guess.